Hello guys. So I've decided to make this video in order for there to be clear instructions on how you can download the software and run it on your computer, and also about what you can do with it. Um, so let's get into it. About downloading, in the GitHub page, and I might also put it in the description for the video, there is actually a Google Drive link. You can see it if you'll scroll down. It's right here, and you can press download, press download anyway, and it will download a zip file. When it's done downloading, I suggest you just go to downloads and do this, extract files. By default it will send it into downloads. You should have it in a file, in like a folder, not a file, sorry. So get in there. Of course you can choose to put this folder wherever you want. As you can see, there is an X file, even has a logo. If we zoom in, yep, just open this file, brainmap9000 v1.1.0, and after I think about 10 seconds it should open if you don't have a Python installed on your computer. Anyway, now that we're here, let me just go over what you can do in here briefly. So first of all, let's just take a wider look. You can zoom just regularly if you hold control and scroll. So it's supposed to be one big figure representing the entire human nervous system. Um, the reality is that currently it's a bit more detailed um, when it comes to the face, and the rest of the body is pretty general, but this will probably change in the future, in future versions I mean. So anyway, let's just dive into the interactive stuff. One thing you can do is you can hover over a given area and it will show you the name of that area. You get it, I think. So second thing you can do is you can view certain nerve pathways, well-known nerve pathways, on the map using this tab here. If you click these buttons, it will show you the pathway. Let's say, for example, the auditory pathway, which starts here in these auditory receptor cells called cochlear hair cells. We can see that going down there to the brainstem, to the rostral medulla, into a little nucleus, which is called the cochlear nucleus. Now, you can also hover over arrows, and this will show you where the arrow begins, where it ends, um, what pathway it belongs to, and its number within that pathway, i.e., what number neuron it is within that pathway. So the second neuron goes from the cochlear nucleus to the superior olivary nucleus. That's why it's showing you 2. There it is, superior olivary nucleus in the pons. So from then it continues to the inferior colliculi, to the MGN, and to the primary auditory cortex. And that's that. So in order to remove the pathway from the map, you just need to click it again, and you see it's white again, it's not active. If it's blue, it's active, and so on and so forth. So I should mention that certain pathways or buttons within this region are a kind of group pathways. That is, they activate a couple of pathways from here. So let's, for example, say, oh, and also you can search things. Let's search the glossopharyngeal nerve. Okay, it has all types of portions. Now let's see. You see, it activates all of its portions. If I deactivate one of its portions, it doesn't show it as active. If I turn it back on again, it does show. So I think it's understandable enough. Another thing you can do is, as you can see, there's also this areas tab over here, which has buttons which allow you to see all of the inputs and outputs of a given area, i.e., all of the arrows which point to that area and all of the arrows which come out of that area. So let's say, for example, I'm interested in the solitary nucleus, and I just take a look. I can see arrows coming in from the tongue, arrows coming in from the body. So let's just view this arrow. Oh, so I can see it's getting information from gustatory receptor cells via the gustatory pathway, the taste pathway. And over here I can see it's getting information from chemoreceptors and baroreceptors via the visceral portion of the vagus nerve. And I can also see it's getting information from other mechanoreceptors. So just by that, I can have some image or be quickly reminded of the types of information that enter the solitary nucleus. And we can also see output arrows to the thalamus, to the ventral posterior nucleus. You can see both VPL and VPM. So yeah, that's that feature. That's the main stuff, I would say. there is this one feature which is really in its diapers, so to speak, and it's this neurotransmitter mapping. There is one pathway which, actually two pathways which this is relevant for, which is the direct and indirect pathway of motor control, the basal ganglia motor pathways. If we turn that on, we can see that um, the arrowheads have colors, and these colors map to certain neurotransmitters. So basically, we can see arrows going down from the motor cortex, the premotor cortex and M1 to the striatum, and also from the SMA. And these are glutamate neurons, as we can see, green. And then we can see from the striatum GABAergic neurons are sent to the globus pallidus internal, red. So, as I've said, still in its diapers, but it could have potential. That's about it. I will probably make another video showing you how you can add your own stuff to the map, or just create new pathways between existing areas on the map. And you don't need to know code for that. Yeah, thanks for watching, I guess.